Crop improvement was the earliest method applied to cross-pollinated crops. It deals with the improvement of crops and production of new crops varieties which are far superior to existing types in all characters. The result of improvement leads to higher yield, agronomically more stable, resistant to diseases, insects, drought, frost, floods, alkaline and saline conditions, improve the quality of produce such as size, color, shape, taste, nutritional value, etc. Different breeding methods are being used in different crops to improve the genotype of crop based on their mode of pollination. Based on the mode of pollination, crops are classified into four types mainly. First one is self-pollination. Self-pollination are generally found in the rice, wheat, barley, oats, chickpea, pea, cowpea, lentil, green gram, black gram, soyabean, linseed, brinjal, tomato, sesame, khesri, sunhemp, chilies, okra, peanut, moth bean and common bean etc. And cross pollination is generally found in corn, pearl millets, rye, radish, cabbage, sunflower, sugar beet, castor, rapeseed, safflower, spinach, onion, garlic, turnip, muskmelon, watermelon, carrot, coconut and papaya etc. Often cross pollination also found in sorghum, cotton, triticale, rye, pigeon pea, tobacco and jute. And some crops are vegetatively propagated as like sugarcane, coffee, cocoa, tea, apple, pears, peaches, cherries, grapes, almond, pineapple, banana, cashew, strawberry, mango, rubber, sweet potato, etc. Breeding methods followed based on the mode of pollination. Self-pollinated crops are plant introduction, mass selection, pure line selection, pedigree method, bulk method, single seed descent method, backcross method, heterosis breeding, mutation breeding, polyploidy breeding, distant hybridization, transgenic breeding, and cross pollinated crops are showing plant introduction, mass selection, recurrent selection, synthetics, composites, backcross method, heterosis breeding, polyploidy breeding, distant hybridization, and transgenic breedings. And third one is vegetatively propagated crops are um, there are following methods are used uh, for crop improvements, plant introduction, clonal selection, mass selection, heterosis breeding, mutation breeding, polyploidy breeding, distant hybridization, and transgenic breedings are used for the crop improvement. Different types of genetic population in plant breedings are used: homozygous and homozygous, heterozygous and homogeneous, homozygous and heterogeneous, heterozygous and heterogeneous. There are the following traits are used for showing the different types of genetic populations in the plant breeding. Different types of genetic populations in plant breeding. First one population, second one brief descriptions and example. Homozygous non-segregating populations and examples are pure line, inbred line, mass selected autogamous varieties and multilines. Second one is heterozygous. Heterozygous are populations segregates on selfing, and examples are F1 hybrids, composites, synthetics, and a clone. Homogeneous, genetically similar population, that is pure line, inbred line, F1 hybrids progeny of a clone. Heterogeneous, genetically dissimilar population, land races, composites, synthetics, and multilines. Combinations are homozygous and homogeneous, genetically similar and non-segregating populations, pure line and inbred lines. Heterozygous and homogeneous are genetically similar but segregating on selfing, F1 hybrids between inbred lines and progeny of clones. Homozygous and heterogeneous, genetically dissimilar but non-segregating populations. Multilines and mass-selected varieties in autogamous species. Heterozygous and heterogeneous, genetically dissimilar but segregating populations, and composites and synthetics. Characteristics of cross-pollinated crops. Each genotype has equal chance of mating with all other genotypes. Individuals are heterozygous in nature, higher degree of inbreed depression, wide adaptability and more flexibility to environmental changes due to heterozygosity and heterogeneity. Cross-pollination permits new gene combinations from different sources. Individuals have deleterious recessive genes which are concealed by masking effect of dominant genes. Focus of breeding crops: population improvement, heterozygosity, quantitative traits oblique characters to predominate and desirable genotype in population, arising new genotypes or gene recombination. These are the following steps used for crop improvements: methods of population improvements or methods of crop improvements, methods with progeny test, initial selection based on the phenotype, final selection based on the progeny test, methods without progeny test, selected on the basis of phenotypes, air to row method, recurrent selection, and mass selection. A brief description of various selection schemes for population improvement. For intra-population improvements, and brief description for improvement within a population. First, mass selection, selection based on phenotype of individual plants. Selected plants reproduce by open pollination. For one sex, inferior plants present in the population are data cell open pollination among the remaining plants. For both bisexual plants, all plants in the population allowed to produce pollen, open pollination without any restriction. Stratified field divided into small plots of about 20 plants each, selection within small plots, open pollination without any restriction, selection usually for unisexual only. Conscious control: plants of a single genotype, single cross inbreed used as check and planted after every 2 to 4 hills for comparison. Check plants, datasil, other plants open pollinate. Selection usually for unisexual plants. Family selection. Selection based on mean of individual plant progenies or families. Half-ship plants within each family, individual plant progeny half-ships that is have one parent, usually the female in common. Year to row method, families produced by one. Open pollination, selection within superior families, no replicated trial, unrestricted open pollination among all the families. Modified year to row, as in year to row superior progenies identified by the replicated yield trial, pollen source and random bulk of all the families. Half-rib selection as the modified year to row but only superior progenies planted in the crossing block and allowed to open pollinate. Modified half-ships used for yield trial S1 families from plants producing superior half-ships intermittent through open pollination. Broad-based test cross half-ship families produced by crossing the selected plants to a tester with a broad genetic base, parental or unrelated, used for yield trial. S1 progenies from plants producing superior half-ship families intermediate, and last one is narrow based test cross as the broad based test cross but the tester has a narrow genetic base. Full sim full sips intermediate, full sips used for yield trial, superior full sips intermediate, S1 progeny is intermediate full sips used for yield trial. S1 progenies from the plants producing superior full sips intermediate inbreds or selfed families produced by selfing. S1 families produced by one generation of selfing used for evaluation. Superior families intermediate simple recurrent selection. S2 families produced by two generation of selfing used for evaluation. Superior families intermediate interpopulation improvement, two populations improvements simultaneously for combining ability with each other. Half sim reciprocal recurrent selection reciprocal with half sim progeny. Full sim reciprocal recurrent selection, each selected plant in the population A is selfed. Each selected plant from A is test crossed with one selected plant from B. The test crossed progenies are evaluated in field trial. S1 families of plants from a producing superior test cross progenies are intermittent. The same is done from B. Requires at least 2 years plant in one of the two populations. Merits of the crop improvement system. Selection is extreme, simple and rapid. Selection cycle is very short and only one generation. Highly efficient to improve, easily identifiable and high heritability characters like, for example, plant height, size of the ear, date of maturity. With proper care of effective to improve yield as well. Most crops have a high additive component of genetic variance which respond to selection. Extensive yields trials are not required as improved strain is likely similar to original population, and demerits are selection based on the superiority is poor basis of selection. Both superior and inferior plants present because of open pollination so reduces the effectiveness of selection. High selection intensity reduces population size, leads to inbreeding, depression. Stratified mass selection, suggested by Gardner in 1961, also known as grid method of mass selection, entire field is divided into small plots, each having 40 to 50 plants. Superior plants are selected in each small plot. Seeds are selected and composited to raise the next generation. Crops, pearl millets, and variety are Babapuri, Jamnagar Zient, AF3, S530, Pusa Moti, all from African introduction. Rye, Brassica juncea, type 11, L16, Thoria, Abohar, yellow sarson, Brassica campestris, T42 and T16. Brown sarson 17 dwarf, 17 medium, DS1 and DS2. Maize T41, T19, Jaunpuri. Cotton, Deshi cotton, C402, C520, C1281, K12, American cotton, 100F, 216F, A19, Castor S20, B1, B4. Year to row method, that is first year original population, plants selected on the basis of phenotype, open pollinated seeds from each plant harvested separately. Second year, small progeny rows grown from the selected plants, superior progenies identified and selected. And third, plants allowed to open pollinate, seed is harvested separately. Third year, same as in the second year. First selection cycle, second selection cycle, third selection cycle. Modification of year to row method. First year, second year, third year and fourth year. First year, plants selected on the basis of phenotype, and second one is open pollinated seeds from each plant harvested separately. And second year, small progeny rows grown from the selected plants, superior progeny is identified. Remaining seeds from the plants selected in first year producing superior progeny is bulked to raise the next generation. Third year, the bulk seed, year to item planted, plants allowed to open pollinate, plants with superior phenotype selected and seed harvested separately. Uh, and fourth year, as in second year. And the second one is, what is the center of origin? The center of origin is a geographical area where the particular group of organisms first originated on Earth, many people believed that centers of origin are also centers of diversity. The crops first crops are cereals, rice, that is also known as Oryza sativa, and it have uh, 24 and 48 number of chromosomes. Center of origin for rice is Southeast Asia, and distribution of species are China, India, Japan, Korea, Pakistan, and Bangladesh. Wild relatives are species are Oryza nivara and Oryza officinalis. Second crops is maize, that is also known as Zea maize. Center of origin are Central America, and distribution of species are found in USA, China, Russia, Canada, and many South Asian countries. And wild relatives Zea mexicana and Zea perennis. Third one is sorghum bicolor. Center of origin is Southeast Africa, and distribution of species are Africa, South and Central India, China, Argentina, Australia, and South and Central plains of US. And wild relatives are sorghum halepense and sorghum drummondi. Fourth is pearl millet. Uh, Center of origin is West Africa, and distribution of species are Africa, India, Pakistan, South East Asia, USA, and Europe. And wild relatives are purpurcum, and pearl. Next is pulses, uh, red gram, that is also known as Cajanus Kazan. Center of origin is Africa and India, and widely distributed in India, Uganda, Kenya, West Indies, and Burma, etc. Soya bean, center of origin China, and widely distributed in USA, Brazil, China, Argentina, and India. Green gram originated in center of origin is India, and widely distributed in India, Pakistan, Bangladesh, Sri Lanka, Philippines, Taiwan, Thailand, Nepal, and southern Asian countries. Next one is black gram. Center of origin is India, and also distributed in India, Pakistan, Sri Lanka, and South Asian countries. Next one is cowpea. Center of origin is Africa, and widely distributed in Nigeria, Niger, Burkina Faso, Ghana, Kenya, Uganda, Malawi, Tanzania, and India, Sri Lanka, Burma, Bangladesh, Philippines, Indonesia, Thailand, etc. Next one is oilseed, groundnut. Center of origin is Brazil, and widely distributed in India, China, USA, Africa, South and Southeast Asia, etc. And next one is sesame. Center of origin is India and Ethiopia, Africa, and widely distributed in India, Pakistan, Africa, China, Mexico, Iran, and Iraq. Next plant is sunflower, and center of origin is North America and widely distributed in USA, Romania, Canada, India, and USSR. Next crop is castor, that is also known as Ricinus communis, and this is center of origin are showing Ethiopia and also distributed in African countries, Egypt, China, India, Middle East, etc. India, Andhra Pradesh, Karnataka, Maharashtra, etc. Some fodder crops are barsim, and barsim center of origin is Syria and is widely distributed in Egypt, India, Pakistan, South Africa, USA and Australia. And second one fodder crops is lucerne, and center of origin is Mediterranean region and widely distributed in Spain, China, Sweden, North Africa, Italy, Switzerland, North America, etc. Next one is vegetable crops. Risgard, that is also known as loofah. Center of origin is India and widely distributed in West Africa, Nigeria, Ghana, Madagascar, etc. Next one is snake guard. Center of origin is Southeast Asia and widely distributed in India, Bangladesh, Nepal, Pakistan, Sri Lanka, Indonesia, Malaysia, Myanmar and China. And next one is bitter gourd, and this center of origin is tropical Asia and widely distributed in India and Indonesia, etc. Next is lucerne, and widely distributed and center of origin is Mediterranean region and widely distributed in Spain, China, Sweden, North Africa, Italy, Switzerland and North America.